Good afternoon, everybody, and welcome to the July edition of the DEBRA UK Research and Health webinar. We are extremely lucky this month to have Dr. Svitlana Kurinna from the University of Manchester, who will talk about RNAs, how RNAs regulate adhesion forces in skin repair and epithelial. Before I pass it on to Svitlana, I'll give a brief introduction. So today she's going to talk about how microRNAs can provide some sort of regulation in how skin repairs or skin adhesion between the epidermal and the dermal layer interface. She is currently an MRC Fellow leading a research group at the University of Manchester, and she completed her PhD in Genes and Development at the University of Texas, and she worked with Professor Sabine Werner in Zurich as well. So she comes in with a wealth of experience. So before I pass it on to Svitlana, just some house rules. All attendees' cameras and microphones are switched off. The webinar will be recorded. Please add your Q&A in either the chat room or the Q&A function, and we'll answer those questions right at the end. And please leave feedback. And if you've got any questions or later on after webinar, please email me on research@debra.org.uk. And on that note, I'm going to pass it on to Svitlana. Over to you, Svitlana.
Hello, good afternoon, everyone. Thanks so much, Agir, for the introduction and for letting me speak here. I know I am probably too much of an RNA person to really be qualified to present the seminar, but it so happened that my microRNA-RNA relationship led me to cell adhesion. And this is why I'm really here, seeking also your input into this really new field that I ran into, having some experience in genes and development, but mostly in the genes, as you will see. And so indeed, if I share my screen, you'll see that most of, can you see it? Sorry, just, yeah, I guess you can. Let me see if.
We can see it, but it's not on slideshow.
Oh yeah, yes, thank you.
Perfect.
Yeah, so as you will see from the data, most of it is on nucleic acids, and this is really my expertise. I really wanted to see the beginning of the process of protein making, and of course the proteins are the ones that are going to make the difference and regulate skin development, regeneration, deposition of extracellular matrix. It's all in the hands of molecules other than nucleic acids. However, at the beginning, it's the DNA and the RNA that regulate how much of what is made and how the signaling is working. So in my group, we started with a very talented master student, Jinqing Zhang, who together with the postdoc, Lalita Thiagarajan, started firstly just collecting the data online while I was buying equipment and trying to establish infrastructure for my group in Manchester. They found that there are plethora of gene expression data, mRNAs and microRNAs profiling coming from primary human keratinocytes isolated from neonatal skin, and the data was really good quality. All that was needed was the analysis to match mRNAs and microRNAs together according to their function, which I will describe in a moment, and just to see how these changes in regulation of mRNA are progressing through simplistic but widely used for its simplicity and information at the same time, keratinocyte differentiation model in 2D, where primary human keratinocytes freshly isolated are considered day 0, and then they are grown to confluency, allowing them to initiate early differentiation roughly resembling suprabasal layer of the epidermis in the skin. And then this graph, or this heat map rather, shows how the changes are not really great if you look at the profile of just 1,000 mRNAs, but they do change from left to right, and mostly they are here sorted by mRNA expression in value that corresponds directly to the number of copies of the mRNA in the cell. So this quick overview showed us that the changes in mRNA and microRNAs are rather subtle, and there are not that many microRNAs that probably make a lot of difference in the skin. However, these changes might be of importance because overall we know that keratinocytes change their protein and lipid composition significantly as epidermis is being built. And so we proceeded trying to match these microRNAs and mRNAs functions. And what we discovered in this particular study was that it's not only the drama maker, microRNA 21, for example, very famous in the skin, or microRNA 205, that are changing expression of mRNAs, but it's also the medium kind of expressed microRNAs like 23 and 29 that impact expression of mRNA, or rather translation of mRNA. And this is what I would like to talk about in this slide, and that is how exactly microRNA is processed and how does it make difference in protein synthesis. So you can see that there are really two major points where processing of microRNAs can impact development of the skin. In the study on the right, this is the study from Ellen Fuchs' lab, you see the pups of mice at just birth. I think it's day 0.5, so just born mice, where essential components of microRNA processing pathway like GGCR8 and dicer that make microRNAs, these short microRNAs, are being knocked out. And at the beginning, or in utero, there is no difference. But as soon as mice are born and start facing oxygen-rich atmosphere, and skin needs to perform its barrier function, the development of it is severely hampered, and these mice die soon after P6. So day 6 is their pretty much survival point, which confirms that microRNAs are essential in formation of normal skin barrier and function. Now, in the lab where I did my postdoctoral training, Sabine Werner was really interested in development of the barrier from the transcriptional point of view. And we were investigating this transcription factor NRF2, which led me to microRNA 29 originally, before I conducted my own study and found that indeed it had a function in the epidermis. So together with colleagues in Sabine's lab, we discovered that if we overexpress this transcription factor NRF2 in the skin of mice, specifically in the epidermis under K5 promoter, we lose this desmocollin protein, DSC2, that is important for formation of desmosomes between keratinocytes. And part of this mechanism is due to microRNAs being activated by NRF2. So when NRF2 is activated, microRNA 29 family, all of it pretty much, is expressed more. And that means that there is less mRNA of desmocollin 2 being made, which results in dysfunctional desmosomes and disrupted skin barrier. Mice that express constitutive NRF2 under K5 promoter and also express more microRNA 29 have a cranky skin that really resembles mice from Ellen Fuchs' lab, but not to the severity of this model. Probably because it's only one microRNA affected plus a transcription factor, whereas in this model, no microRNAs are pretty much can be made through missing DGCR8 or dicer. And so I decided to pursue function of microRNA 29 because I wanted to see what exactly it does other than targeting desmocolin. The reason for that is microRNAs work through complementary binding to mRNA through a very short seed sequence. It's only 6 to 8 nucleotide long, and if you predict this sequence, or if you look for it simply in the 3' UTR of mRNA, you'll find many mRNA targets. In fact, we predicted around 2,000 mRNA targets for 29, microRNA 29. That's too many to really be functional, simply because stoichiometry inside the cell would not allow microRNA 29 bind all of its targets. Besides, not all of them are expressed, which is exactly what we tried predicting here when we looked at microRNA's profile versus mRNAs. Altogether, this led me to realize that I either need to continue this matching bioinformatics game and then functionally somehow validate these pairs between microRNAs, or focus on one microRNA, which is kind of in front of me, and do a biochemical study to confirm, or rather really fish out, targets that this microRNA doesn't only predict it to bind, but actually binds inside the cell. And I've chosen this path really because there was a technology developed in the lab of Jonathan Hall across the street in Zurich that allowed me to firstly detect this microRNA specifically by the probe, by in situ hybridization, and show how it is expressed in the epidermis. In situ hybridization is nothing new, but detecting it with a specific probe for a microRNA is tricky because, again, the complementary binding can be random and unspecific due to short sequence of microRNA. So we showed in this study that microRNA 29 is largely missing in the basal layer of human epidermis, and that is because in the suprabasal layers it's upregulated by a master regulator of epidermal development, P63, joining forces with NRF2, activating microRNA 29. And then I've developed this hypothesis where I just took layer by layer epidermis in this prediction algorithms and created this kind of maybe too complicated, but somehow explainable for at least in my mind, diagram, probably too colorful as well, where I'm trying to show that gradual changes in mRNA levels in the epidermis, let's say those regulated by microRNA 29 or other microRNAs, will have opposite gradient. I mean, this is nothing new, because if you think microRNA suppressing mRNA, the gradient of each of them should be always opposite. But the point is for microRNA 29 is that as it is expressed gradually upward in the epidermis, the repression of mRNAs is also happening gradually, and this is a key for healthy epidermis. So we hypothesized that in the absence of microRNA 29 in the basal layer, keratinocytes would then retain the ability to grow and repair through stem cell properties like basal adhesion and extracellular and intracellular signaling that is linked to it. So in effect, when keratinocytes start differentiating, microRNA 29 kicks in and starts repressing the basal functions. So the suprabasal differentiation is due to the loss of basality through microRNAs. And then Jonathan Hall's team helped me also with this amazing technique called microRNA clip, where probe for microRNA is specifically synthesized and labeled to allow, by sorrelin and biotin, to allow binding of microRNA to its mRNA targets cross-linking it, kind of locking it with sorrelin, and then pulling on argonaute and biotin tandemly, so in tandem, where AGO2 is a complex that microRNA binds and drags mRNA into, is isolated first, and then microRNA 29 cross-linked to its mRNA targets is isolated, the second step. So when this probe is transfected into keratinocytes or any other cells for that matter, it can fish out microRNA 29 bound mRNAs very specifically. And it is a real life type of situation. It's not prediction based because of many step washes, to put it simply. Only cross-linked bit between sorrelin and microRNA binding site on mRNA will remain after this tandem pull down and will get into RNA sequencing. And so when you do RNA sequencing, you end up with lists of targets of this particular microRNA in the cell line or in a cell. In our case, it was primary human keratinocytes and primary human fibroblasts isolated from skin biopsy. Of interest, can be anything really. I think I'm covering up part of my data, or can you see it somehow? Let me try. Yeah.
No, we can see it.
Oh, you can see it all.
Yeah.
Okay. Okay, sorry. So, yeah, because this is kind of important. I covered up the exact bar that I wanted to show, at least on my screen. So this is the result of microRNA clip, where human follicular keratinocytes were isolated, interfollicular keratinocytes isolated in parallel with dermal fibroblasts, transfected with microRNA 29 probe labeled with sorrelin and biotin, cross-linked, tandemly purified, RNA sequenced. I make it sound so simple, but Lalita, Dr. Tyagarajan, spent a year and a half doing this, just after all the optimizations. And we're still, I'm afraid, following up on her experiments because the analysis proved to be even more laborious than I expected. But what really jumped almost immediately after gene ontology and enricher algorithms were employed was that all cell types, all three, majored in cell adhesion function. I honestly wanted to get something else just because I thought can't be only adhesion that this microRNA targets. And now thinking back, I realized, well, of course it can be only adhesion because probably microRNAs are specialized in what they do. They don't just randomly target everything they can complementary bind, but there is some kind of synchronized expression of microRNAs with mRNA targets that goes gradually to regulate developmental and regenerative processes. And only when it's misregulated in a disease, you will get this balance disturbed, and it will be either too much of microRNA or too little of it, with corresponding changes in mRNA. So as you see from these graphs, there are other functions for microRNA, but because adhesion, again, was the one in front of my eyes, I decided to follow up on it. And this is where it took me to the new field that I'm talking about from this slide on, with perhaps a bit less confidence, as it is very fresh unpublished data that I'll be happy if you criticize. So the atomic force microscopy is really not the method of choice when one wants to study cell adhesion at first. The reason I wanted to do it because I did not know really what else was kind of a golden standard in the field up in front. And when I was just reading about it, AFM jumped at me, maybe because it's letter A, and it's somehow listed first. but perhaps more so because I've heard of it vaguely in conjunction with matrix and adhesion analysis. And the instrument was available at the core facility at the University of Manchester. I had really good specialists helping me. So I decided to go ahead with it, and it is very interesting technique. So the probe, that is a very sensitive cantilever, can pick up single cells, like little balls, and then touch the surface, for example, coated with fibronectin, let adhesion receptors get involved with fibronectin by gently pressing it down, like on step 2, and then lifting it up, it can record piconewton force that takes every receptor to disengage. And this is what this graph is showing. So the detachment force on the negative y-axis is actually the interesting bit. You see that it goes in kind of a stepwise manner, like a ladder, or rather a staircase. So it's really reflecting how many integrins, for example, are engaged with fibronectin. And as you tear this all up and they disengage, you get these little jumps that create steps. So what I did next was I took human keratinocytes, I added more microRNA 29 to them, and you know keratinocytes, when they're healthy, they can be very sticky. They really adhere well to coating or to the matrix. And then by doing AFM on control cells and microRNA 29 transfected cells, we recorded this difference. So we first confirmed that fibronectin was making a difference to keratinocyte adhesion compared to BSA, which is just a control coating. And then, as the blue bar shows, we saw that this adhesion to fibronectin was significantly decreased when we overexpressed microRNA 29. So this was the first and so far the most solid in my mind confirmation that microRNA 29 directly regulates adhesion strength of human keratinocytes. And then I did, I'm afraid, too complicated of a setup, which I will do my best to explain here. I took primary human keratinocytes and transfected them with antisense oligonucleotides that would inhibit microRNA 29. So it was working against activity of microRNA 29, presumably making cells attach better, and as we showed, also behave kind of more pro-growth and more like basal keratinocytes. So the antisense oligonucleotide was labeled with Cy3 label, and you can follow the transfection and adhesion at 10 minutes and then 24 hours of the growth of the cells on this panel here, showing that oligonucleotide bound to microRNA 29 and inhibiting its activity is retained by cells that continue to grow. And then we separated these cells into fast attaching cells and slow attaching cells by simply removing the fraction of cells that did not adhere at 10 minutes, and we call them fast. And we call them fast ABC because ABC denotes antisense oligonucleotide against microRNA 29 A, B, and C sequences that are found in vivo. And then we did RNA-seq. And the analysis was done by comparing keratinocytes that have miR29, that do not have microRNA 29, that are quickly attaching and that are slowly attaching. And to say that microRNA 29, to confirm that microRNA 29 had an effect on adhesion of cells, we did this also rather simplistic analysis that I just counted cells in a cluster to show that they adhere better by other means when microRNA-29 is inhibited. If you also notice an interesting thing in the ABC cells, nucleus to cytoplasm ratio is different. I didn't quantify it, but I think it's a sign that these cells may be more like basal stem cell-like, that microRNA-29 inhibition supports this basal state, but this evidence needs further investigation. This is the result with the enricher analysis of RNA-seq in fast and slow cells with or without miR-29. So if in control cells, all these differently colored balls and numbers denote molecular functions involved in adhesion, you see that they get significantly enriched when we remove miR-29. And that is expected now, because we know there is a function and we know miR-29 inhibition would release mRNAs. So mRNAs should bounce up, and they did. Moreover, when we looked particularly at these mRNAs, we immediately noticed a couple of obvious candidates for pro-adhesion, and one of them was collagen 4. So cells were expressing much more collagen 4 when we removed miR-29. And when we did it with dermal fibroblasts and collected the conditioned medium from them, and then plated keratinocytes and fed them with this medium, we noticed that keratinocytes grew much better when miR-29 was inhibited with the ASO in the fibroblasts, conditioning the medium for these keratinocytes, suggesting that there is a paracrine signaling on top of autocrine signaling, feeding keratinocytes with something that allows them to grow and attach better. Moreover, there was much more matrix deposited by dermal fibroblasts when we removed miR-29. That is not exactly surprising because it was reported that miR-29 regulate collagen synthesis. So by removing miR-29, one would expect more collagen deposited by fibroblasts. However, we are pretty sure by now that it's not only collagen, that there are other factors that are released by miR-29. And because I cannot show you, of course, all the targets that I would like to follow up on, I will only focus on the few that directly relevant to adhesion. This is something we tested now in 3D cultured cells, where we put keratinocytes on top of fibroblast scaffold and let them grow and differentiate for up to 11 days. So in this left-hand side panel, you see two control rows that would be NSM and NSA. So the top two rows are showing the control kind of situation where control oligos were transfected. And then the two bottom rows are showing differences in they show difference in target deposition, but microRNAs would be inhibited or upregulated. And I've actually misaligned these errors, and it's really misleading now. So I am going to move them quickly down. Sorry, yes. So now it's in place. So if you now just focus on the two bottom rows, you will see that when we overexpress miR-29, the skin equivalent is not as thick, and there is some integrin alpha-6 deposition, but there is definitely thicker equivalent and more integrin alpha-6 when we repress miR-29 with ASO. So when we overexpress miR-29, we lose some integrin alpha-6. When we remove miR-29, we release integrin alpha-6 and also beta-1, but it wasn't significant when we quantified it. It's probably not so profound, but both of them are direct targets of this microRNA, and I do not see why they would not be directly repressed. So finally, I would like to move on to in vivo data, which is my favorite and also the most troublesome to interpret, because I always feel there is too big of a jump from even primary cells that are cultured, isolated freshly from human biopsies and a mouse model. But there is really nothing to fill this gap other than doing 3D models at the moment, which are great, but also, of course, missing all the systemic response to skin damage. So in my license, I am allowed to do two 6 millimeter biopsies on mouse dorsum, and then I can let wounds heal for a number of days and collect them at different time points to investigate how reepithelialization proceeded. And so when I imported miR29 knockout model from Belgium and performed this study, I will show you here a part of it where we measured distance between wound epidermal edges as they regenerate and kind of go in to close the wound. Of course, in mice, this contraction factor is really big because they can just shrink their skin and bring edges of the wound closer together, but you can still just about observe reepithelialization process as it happens in humans. So we detected a difference between knockout reepithelialization. So when miR29 is removed, we see just a little bit better reepithelialization and also better formation of small blood vessels in the granulation tissue, allowing wounds heal. And when we look at microRNA-29 using this in situ hybridization with the fancy probe, we can again see how it's missing from the basal layer, and it is really expressed in the immediate suprabasal layer up in wounds as well. And if you look at day 5, when some wounds are already closed in mice, you can tell that removing of miR29 by the knockout in a mouse results in somehow thicker wound matrix. For the lack of better word, this Herovici staining is just really interesting. It shows you how there is more matrix, more immature collagen going into mature, but doesn't exactly allow you to quantify it and put your finger on the mechanism. However, the result speaks for itself. A better reepithelialization of knockouts is really strong indication that removal of miR29 or its suppression could be beneficial for skin repair. And this is what I've been really struggling to do for the past 2 years, I would say, or rather my former PhD student and very talented postdoc who finally finished his training, really tried achieving, was to knock down miR29, so not to use genetic knockout, but inject these antisense oligonucleotides, ASOs, labeled with Cy3 into the wound and see if we could in situ just get this matrix form better and reepithelialize wounds better. We have very partial success with it, and part of the reason is that no matter what kind of chemical modifications we introduce into ASOs, we just can't get them go into the epidermis. They go everywhere in a wound, but not into epidermis. So Connor, this postdoc who was conducting this study, learned laser capture microdissection on wound sections. And because they were labeled, he could cut different sections, compare them to each other, and compare non-specific ASO to microRNA inhibitors. And he saw that microRNA levels were going down, but only partially and only in the sections where I think mostly fibroblasts are present. So we couldn't achieve the knockdown where we wanted, which is the epidermis. And for the epidermis, therefore, I still rely on the data from knockout mice. But it gives me a window of opportunity to at least consider that once I get the data on profiling microRNA expression in epidermolysis bullosa samples, perhaps we could think about delivering microRNA-29 inhibitors into blistering wounds. And this is where I may now transition into this skin disease that your community studies, because I don't know much about it, and this is where I need help. So I've been in touch with Dr. Peter Acker from the Netherlands, and he told me, yeah, let's take a look in EB samples for your microRNA, but could you design a trial for it? So I have a number of patient samples. How many do you need? I don't know. Well, I would say as many as you can give me, but I actually don't know how to design a trial with human samples. Ask me how many mice, I'll tell you, but with humans, I really don't know what's the best way of doing it. So if any of you could get in touch with me about this, I would be really thankful. So I would like to make these kind of cartoonish conclusions to microRNA 29 function in the skin, simply because I actually don't know exactly what it does to adhesion. I just know it does something to it, and it does so in human keratinocytes and fibroblasts as they talk to each other. So in wounds, as shown on the left, microRNA 29 in the basal layer of keratinocytes is expressed below the point of detection, but it's immediately expressed in the suprabasal layer, and we've showed the mechanism. It's through P63 and NRF2. It then, what we think is, downregulates markers of and functional mRNAs proteins in that would represent basal keratinocyte phenotype, which allows keratinocytes to be more flexible and migrate to close the wound. And if we remove miR-29, this process is enhanced because there is nothing to prevent more keratinocytes behave more basal-like. Inhibition of miR-29, or its knockdown, also at the same time does something to endothelial cells and fibroblasts in the granulation tissue. We see enhanced development of blood vessels, so that feeds back into better wound closure. And then on the right-hand side, I'm showing how this is playing a role when we look at keratinocytes and fibroblasts through microRNA clip. And we show that miR-29 directly binds mRNAs in ephrin, ERK signaling, TGF-beta and catenin pathways, and of course, integrin interaction with laminin. We also have soluble factors affected in the dermis, as conditioned medium experiments show, and one of them being SPARC, and collagen 4 deposition by both keratinocytes and fibroblasts is changing in response to miR-29. So all in all, I would like to conclude by this most simple cartoon that we used as a little snapshot abstract for our most recent publication, and that is that somehow when you inhibit miR-29, you get an additional little adhesion. Thank you very much for your attention. I would like to actually thank the people as well, and my funders, so MRC and Smith and Nephew, the group of Sabine Werner, and my own group, of course, all the people who are changing so fast. I'm missing many already, who moved on with their careers, my collaborators, and the core facility of the University of Manchester. And thank you all again.
Thank you, Svitlana, for an excellent talk there. Oh, sorry, I'm slow. Sorry, there you go. Sorry about that. No, thank you, Svitlana, for an excellent talk there. I certainly learned a lot about miR-29 and the impact it has on cell adhesion. I'm going to jump straight into a question here while everybody's thinking about questions and coming up with what they could ask you. Okay. My question is, could knocking down miR-29 potentially be a universal treatment for all EB types. So when I say all EB types, obviously that's EB simplex, junction, and dystrophic EB. You won't resolve the underlying genetic problem, but you could have better wound closure.
Yes, so I think potentially knockdown of miR29 could benefit all types of EB because microRNA is having function in the dermis as well. But we've had this discussion with you when I start talking about this too boldly, I get this paper flanked at me where a good paper shows that in recessive epidermolysis bullosa, there is very little microRNA 29 detected. And I think the reason is that this intervention has to happen much earlier when the skin already scars. There is a lot of TGF-beta, and it's a known transcriptional suppressor of miR29. So it just doesn't let it be expressed, but it's anyway too late to release any of its targets because they're all already messed up.
So you think if miR29 is a potential treatment option, it should be really early intervention.
Yeah.
I'm thinking more children at a young age, because by the time you've got to adults, you've already got that fibrosis, that the scarring, and any potential treatment is too late.
However, I would throw in this thing that there is a clinical trial for microRNA 29 mimic, which was reducing fibrosis in some scleroderma type of disease, so not EB, but there is a potential for the opposite way. So if there is too little miR29 and it's too late already to prevent scarring, you can still target fibroblasts that started just depositing too much matrix with miR29 and help them to shrink back.
Okay. So it's almost the opposite. You're getting rid of the scarring or the fibrosis
with putting more miR29.
Yeah.
But if we intervene earlier, I think removing miR29 might be the beneficial.
Perfect. Yeah. Do you know if there are any? Obviously you've just mentioned one clinical trial for putting in more miR29. Do you know if there's any ASOs out there that will knock out miR29? I know you've got some ASOs within the lab, but I was wondering if anybody translated that to a clinical trial or potential treatment.
Not that I'm aware of.
No, perfect. And again, we've got a couple of questions coming in. So I'm just going to read them out. So we've gotten saying, thank you for a very interesting talk. Have you considered modified herpes virus to deliver molecular reagents to mouse wounds as used for RDEB therapy?
I know where it's coming from. Thank you for the question. Yes, it would be amazing if. Yeah. I'm just too premature for it. Should I say no? I have not. I would dream about it, but at the point where my lab and research is, I would love to talk about this, but I cannot do it yet.
Yeah, that's okay. Yeah. And another question came is, how would a treatment get the miR29 ASO into skin cells?
Yes, so this is the trick with modifying these single stranded oligonucleotides to stabilize them, because usually they just degrade, and then add another modification to make them penetrate the cell. Again, partial success, but it's a success nevertheless.
Yeah. Do you know what? I think talking to Peter van der Hacken, he would give you some advice on stabilizing ASOs, and also trying to get them into the skin cells.
You mean keratinocytes?
Yes.
Oh yeah.
Peter has a lot of experience in that, and good experience.
Peter Acker, right?
Yeah.
Yeah. Mm-hmm.
Yeah.
Thank you.
I guess. So another question's come through. Are there any other treatments that upregulate collagen 4 that could be tested on EB mice?
Ooh, I don't know. This is why I was so excited that we removed miR29 and col4 jumped up, because I don't know any other ways to upregulate it other than injecting a peptide.
Okay. I've got another question. So I think I might have missed it, but early on when you add in the ACEs into the keratinocytes and you've got reduced miR29, you then took the media out and put them onto other cells, and you show that actually those other cells also have better cell adhesion. Could that possibly be a treatment? So apply the media onto wounds with sites.
Yeah, so this was fibroblasts with knockdown miR29 and then the conditioned medium. What we really need to do is to get the conditioned medium through proteomics, because I need to know what's in it before we go any further.
Okay, because I guess that could be an alternative way of getting the same effects of knocking out miR29 rather than trying to use the ASOs if you've got.
Yeah, no, that's a good idea. That's a very good idea.
You know, so I could potentially see that as like a topical treatment, topical gel treatment, which might be easier, and like I said, it potentially could be used for all EB types.
Yeah, yeah, exactly.
Yeah. I don't think we've got any more questions. So if anybody's got any questions, please put them in. You've got a few more minutes before we close the webinar.
Thank you for the reference. Someone had sent, was it Enrique? Yeah, to host and panelists. I see it. Thank you for picking up the reference. Yes, my contact is my first and last name, and then at manchester.ac.uk. I'll just type it in.
And then we've just had a question through the Q&A function. They said, can the treatment be delivered orally or topically?
Orally, probably the oligonucleotides will not survive even with all their modifications. So that's all in chemistry. Depends. Topically, was it topical? Yeah. Topically, yes, that's exactly what we're testing in mice. So yes, they are delivering into wounds better than I thought, just not into the epidermis.
Okay. So Svitlana, you just added your email address in the chat function just to the panelists. Should I share that with everybody on?
Yeah,
okay.
Yes, please.
So I'll do that now. Okay, so that's done now. And then somebody has put in a question, does it work with laminin?
Yes, so we published this last year with laminin confirmed as a direct target of miR29 in vitro. In vivo, it's very interesting. I didn't show this data, but in wounds, we get more laminin 332 deposited, but not where we would expect it. So we thought it would be, basal layer getting thicker. Instead, it's all in the wound matrix, and we think this is what somehow promotes angiogenesis. We saw it also in patient samples in the few wounds that I could get from chronic and trauma wounds. So yes, the short answer is yes, but I'm still not exactly sure if the mechanism is direct. It doesn't really matter if it's indirect as long as it's beneficial. So yes.
Well, that's it. I think as long as the wounds close better.
Yeah.
Because fundamentally, that's what everybody wants. It's that clinical benefit and that quality of life. Okay, perfect. We'll give it a couple more minutes before we close the webinar. I guess question for you, Svitlana. Where do you see this work leading to? What's your ideal scenario?
Yeah, so my ideal scenario is to use my expertise, at least for now, to get the function of miR29 and adhesion nailed at the genetic and molecular mechanism level. And this is why I want to cross the knockout mice to EB mouse model to see if it helps. And then, of course, move on with the help of professionals into some sort of preclinical stage.
Perfect. No, and I think that's what people with EB want. They would love to see more clinical trials in EB, and hopefully that will lead to treatments. We just had a question that's come in. It says, in our DEB mice, miR29 is decreased and was related to progressive fibrosis. Yeah. Could this represent a contradiction?
Yeah, so I think it's not a contradiction. It's just really two different timelines where when fibrosis has already taken over, for whatever reason, there wasn't enough microRNA 29 to control it, and fibrosis took over. What it means is that fibrosis is now going to suppress miR29 and feed back into fibrosis by a very clear and very powerful genetic mechanism where TGF-beta activates SMAD, and SMAD just sits on the DNA where miR29 gene is regulated and doesn't give it a chance to suppress fibrosis back. So I think it's the timing.
Perfect. And then we've got another question that said, thanks for the talk, I've learned a lot. I'm curious how to balance the increased wound healing of downregulating miR29 with critical roles it plays that might be affected given its multifunctional nature. I guess this is the tricky balance between when do you knock down miR29 and when do you try to upregulate it. It's a bit like what you said earlier, in earlier intervention is better, but much later intervention, once you've got all this fibrosis, once you've got all this suppression, actually increasing the amount of miR29 might actually be better in that situation.
And the good news is that you can do either or because both of it are nucleotides. It's just the way the sequence goes, right? It's purely in the sequence, but as far as approach, it is possible to do it both ways.
Yeah, perfect. I think that's the last question that came through. So, and on that note, Svitlana, I want to say really thank you very much for an amazing talk. I've certainly learned a lot today.
Thank
you
for
that. No, thank you. And just to let everybody know, so we normally have a monthly webinar. August, we will be taking a break for the summer. So I wish you all a really good summer, but we'll be back in September at the usual time of first Wednesday of each month at 1 p.m. We will share the details of the next webinar shortly, but please do come and register. And before I close the webinar, again, one more time, Svitlana, thank you very much for an amazing webinar. Thank you, though.